Enzymes are proteins that speed up chemical reactions in the cell. A special region on the enzyme, called the active site, has a shape that fits with specific substrate molecules. An enzyme works by binding to one or more specific molecules called reactants or substrates. Binding occurs at the active site. The enzyme and substrates form an enzyme-substrate complex. The interactions between the substrates and the enzyme stresses or weakens some of the chemical bonds in the substrates. These stresses encourage a link between the two substrates, leading to the formation of a different molecule. As a result of the chemical interactions within the active site, a new product is formed. The product is released from the active site. The enzyme assumes its original shape and is free to work again. Although this reaction has specifically illustrated the formation of a single product from two substrate molecules, other enzymes catalyze the formation of two products from a single substrate. An example of how enzymes function in the body is from the enzyme sucrase. Sucrase resides on the surface of the microvilli on the intestinal epithelial mucosal cell surfaces. This animation presents a graphical representation of the way that sucrase catalyzes the hydrolysis of the common disaccharide sucrose, which we know as table sugar, into its component monosaccharides, glucose or blood sugar, and fructose or fruit sugar. Hydrolysis is accomplished because when the sucrose molecule binds to the active site of the enzyme's configuration is changed so that the oxygen bridge between the two monosaccharides is exposed to water molecules in the solvent. This exposure permits a water molecule to actually break the bond, the oxygen bridge, and attach the components of water, an OH to one of the monosaccharides and an H to the oxygen, which is still attached to the second monosaccharide. This effectively cleaves the bond between the two monosaccharides and converts the disaccharide into two separate sugars. Once this is accomplished, the enzyme's configuration is changed back to the original shape. The two monosaccharides float away, and the site becomes available for another sucrose molecule to bind, change the enzyme's configuration, and be hydrolyzed. This action can be repeated many times until the enzyme becomes denatured, is inhibited, or just wears out.